As I mentioned earlier, the second line of defense includes processes, phagocytosis, inflammation, and fever. So the next few video lectures we'll be talking about those processes. Now we've already discussed phagocytosis. We talked about that back in chapter 11. And if you've had your anatomy and physiology classes, you've talked about phagocytosis there as well. So as I mentioned earlier, generally the goal of your immune system is to get things phagocytosed. Now, your phagocytic cells, the main ones, are neutrophils and macrophages. So those are your key phagocytes. Keep that in mind. And generally, they're very good. And we'll talk about how this works. They have ways of attaching to many different kinds of microorganisms and going ahead and engulfing them, performing phagocytosis, and destroying them. So the vast, vast majority of microorganisms that are encountered by your body get phagocytosed, no problem. As we learned about in chapter 11, though, those professional pathogens have ways of getting around phagocytosis early during infection. And we'll see when we get into chapter 13, one of the main goals of your specific immune system, your third line of defense, is to go ahead and get those pathogenic microorganisms phagocytosed anyway. So in general, now not everything, phagocytosis is not the only way that your immune system kills microorganisms, but it's generally the main goal. It is a process of the second line, but it's also important for the third line, as we'll see. And again, your professional phagocytes include the monocytes in the blood that become macrophages or dendritic cells, and then also your neutrophils. All right, so just kind of a review of the key steps of phagocytosis. The first one is chemotaxis. So if you remember that term from earlier in the semester, that is basically following a chemical trail or a trail of chemical breadcrumbs. So here we've got some bacteria. Here this looks like a neutrophil. And the neutrophil, bacteria are releasing bacterial chemicals. Phagocytes have receptors on their surfaces that will attach to those chemicals, and that will guide the movement of the phagocyte towards where those microorganisms are located. So chemotaxis comes first. Number two is adhesion. Okay, so if you focus in here on this part of the diagram, we've got some rod-shaped bacterial cells here, and they're showing you there's some molecules on the surface or markers on the surface. Your textbook is using the term marker quite a bit. And your textbook is labeling those as PAMPs. PAMP stands for pathogen associated molecular pattern. Pathogen associated molecular pattern. So basically what that means is it's a molecule on the surface of the microorganism that you don't have on your own cell. So it's some sort of non-self molecule, but it's something you would find on many different kinds of bacteria or many different kinds of fungi or many different protozoans, things like that. Those are PAMPs, because PAMPs need to be things that help your phagocytes find microorganisms in a non-specific way. So it's not going to be some kind of molecule you would only find on a particular strain of Salmonella bacteria. It's going to be something you would find on lots of different types of bacteria, something that's more non-specific. So that's adhesion. So the phagocytes have receptors called pattern recognition receptors, and those attach to the PAMPs on the microbes. Then the poor little microbes get engulfed, and they're brought inside a phagocytic vesicle. So we talked about that in chapter 11. That's a membrane-covered compartment. As that enters the cell, that's now called a phagosome. Then you have these lysosomes, and that's not the same thing as lysozyme. A lysosome is a membrane-covered compartment that's full of many different kinds of digestive enzymes. And that lysosome will merge with the phagosome, and now you have a phagolysosome. Now the poor little microbes get digested by all those enzymes. And some of the enzymes that are in there, lysozyme is an enzyme you would find in a lysosome. Lysozyme breaks down the peptidoglycan cell walls of bacteria. DNase to break down DNA, RNase to break down RNA, proteases to digest proteins. And so all those enzymes are going to break down the organic molecules that make up cells, that make up microorganisms. So this is also what's going to happen, by the way, when if you have macrophages that engulf red blood cells, or your dead cells that need to be cleaned up and broken apart. Same kind of process is going to take place. So those lysosomes are going to have various digestive enzymes that will help break down any kind of cellular stuff that gets engulfed. Notice also that these lysosomes contain reactive oxygen products. Superoxide, hydrogen peroxide, singlet oxygen, hydroxyl ion. Those are all some of the nasty oxygen-containing compounds that we talked about back in Chapter 6, where we were learning about how aerobic, any living thing on the planet that lives in the presence of oxygen gas has to have enzymes that will detoxify these types of things. Well, here your phagocytic cells are deliberately producing these nasty reactive oxygen-containing compounds to help kill and destroy and break apart the microorganisms that have been engulfed. So that's kind of interesting. Actually, it's not on here listed in your textbook, but your phagocytic cells actually make some hypochlorite, and hypochlorite is the active ingredient in bleach. So your phagocytic cells actually make very minute quantities of bleach to help destroy these engulfed microorganisms. After the poor little microbes have been broken down into their little component parts, the phagolysosome travels back to the cell surface, and all that cellular debris gets released or eliminated. So these are your 7 key steps of phagocytosis. Just take a look at that table in your textbook for a really good overview. Got a link here in the full PowerPoint to a YouTube video you might want to take a look at, which is either I think it's a neutrophil that is following a bacterial cell that's kind of bouncing around and through chemotaxis, and then you eventually see the neutrophil capture the poor little bacterial cell and it gets engulfed. So a little bit more about these PAMPs. So we said molecules that are present, or markers present on the surfaces of microorganisms, things that you would find generally on many different kinds of microorganisms. Those are called PAMPs. It stands for pathogen-associated molecular pattern. They don't necessarily have to be on pathogens. They can be on non-pathogenic microorganisms as well. And those attach to PRRs, which stands for pattern recognition receptor, on your phagocytic cells. Okay, and there's a diagram of that happening. PRR would be sitting on the surface of a neutrophil or a macrophage or a dendritic cell, and the PAMP in this case is the peptidoglycan cell wall of bacteria. So peptidoglycan is a good example of a PAMP because it's going to be a molecule you would find in bacteria, but you would not find, it's a non-self molecule. That's not a human molecule. So that's going to be a good thing for your phagocytic cells to have receptors to attach to. Some other example PAMPs, lipopolysaccharides. Remember, you find those in the outer membrane of gram-negative bacteria. Lipoteichoic acids are found in the cell walls of gram-positive bacteria, if you remember that from Chapter 3. And there are various yeast surface molecules that have been identified. There are viral molecules. There are protozoan molecules that have been identified. Some of this is kind of a newer area, so more and more of these PAMPs and pattern recognition receptors are being discovered all the time. Sometimes the PRRs are called toll-like receptors, or TLRs, so you may see that terminology used as well. Now, when we get into Chapter 13, we're going to be talking about antigens, which are also markers, complex molecules that are present on the surfaces of microorganisms. But when we're thinking about antigens, we're thinking more about molecules that you specifically find on some particular strain of influenza or some particular strain of salmonella bacteria, or on Ebola virus, for example. They're very, very specific molecules that may be unique to a particular type of microorganism. PAMPs are things like this that you would find on many, many, many different microorganisms. So PAMPs are considered to be markers that are used as part of your second line of defense. All right, so keep phagocytosis in mind because it's going to come up again as we continue through chapters 12 and 13. The next video lecture, we're going to talk about inflammation, or the inflammatory response, as a process of the second line of defense. And again, most of that should be a review as well from this class and from your anatomy and physiology classes, but we'll cover a little bit more detail now that we're learning more about some of the cells that are involved in inflammation.